Log on, tune in, find out. Another good idea from Cambridge.
And you can see the title of the talk. I will talk about two types of problems today. They're closely related, but they're a little bit different. The first type of problem is adaptive optimal design for dose response, dose ranging studies. Second type of problem is about optimal designs for population pharmacokinetic, pharmacodynamic studies. And by discussing the second type of problem, I will sort of bridge what we discussed last week at the last week workshop, especially the last day on Friday, with what's discussed today. There is a key common component in both problems, and this is the theory and algorithms, the numerical procedures of optimal model-based designs for nonlinear models. There are differences, and those differences actually exist only for today, because in one group of problems we are discussing adaptive designs which require a built-in estimation module. In the second group of problems we are not doing actually estimation, we are doing only design, but the peculiarity of the second group of problems is the nonlinear mixed effect models. Optimal design, what we are trying to select or optimize. When we consider dose response or dose ranging studies, we are looking for the best, and I'll talk what I mean by best doses, how many doses to select per individual patient, and we try to find the number of patients which are enrolled in this study. When we are doing PK/PD studies, we are trying to find the best sampling times. By best, I mean the most informative. We also want to find the best optimal number of sampling times per patient, given our restrictions. And again, we want to find the number of patients enrolled. What's the number which gives us enough information? Optimal designs. What we mean by optimal? Optimal with respect to precision of parameter estimates, because today we will be discussing some parametric models. So the quality of design will be measured by the quality of estimation, and this is in some sense the size of the variance covariance matrix of parameters. So, and the goal is to find the most informative either doses for the first type of problems or sampling times for the second type of problems. As we all know, both for design and estimation, the key is the derivation of the Fisher information matrix of a properly defined single observational unit. And our single observational unit is our individual patient. The models which I discussed today are, for dose response, I'll give some examples with popular Emacs, which is multi-parameter continuous logistics, and I give some examples with binary logistic models. For PK/PD, it will be compartmental pharmacokinetic models, which are described usually by systems of ordinary differential equations, which may be linear or nonlinear. And I'll give some examples of PD models. Again, it may be an Emacs model, which is very similar to what we have here, or it may be a system of ordinary differential equation too. I'll show you some examples of software tools which were developed over the years in GSK. These are GUI-based standalone applications which were written in MATLAB. Why MATLAB? First of all, it's a very nice programming tool in my view. Second, it allows for creating standalone executable files, so there is no license required for the end user. And it's very easy to create a graphical user interface. Now I briefly go through the standard song. Valery yesterday introduced a new word. Now he's using 8-step dances when discussing optimal design. I will still be using song, and it probably doesn't have 8 steps. It has less than 8 steps. So when we have the expression for the information matrix, then we use independence of patients, which are put either on a dose X on a sequence of sampling time. we create the information matrix of our design. We know that for good estimators, the variance is inversely proportional to the information matrix. And then we work with the normalized information matrix, normalized per observation. So we work with weights, trying to construct the optimal design, not with the numbers n. And we work either with the normalized variance covariance matrix or normalized information matrix. And the criterion of optimality is some scalar criterion which depends on the variance covariance matrix. We try to minimize it with respect to weights, which have to be non-negative, and with respect to admissible axis. So we have to properly define the design region. And the design region is either the admissible doses for the first group of problems or sampling sequences for the second group of problems. And one of the most popular criteria is d-optimality, the determinant of variance covariance matrix. We have a nonlinear problem, and we start with locally optimal designs. We need a preliminary estimate of our parameter. The numerical procedures are based on generalized equivalence theorem, and this equivalence theorem creates the major steps for the algorithms, what we are using, so-called first order Fedorov-Wien algorithm. I have this picture which I borrowed from last week because I thought that Ben Torsney will be here, and we had some discussions about the geometrical illustration of different criteria. But since Ben is not here, I'll skip it. I'll just, if I have time, and if you have further questions, I'll comment later. As I already said, for the first group of problems, we are not stopping at finding locally optimal designs. We are creating adaptive designs. And for the adaptive designs, we have to combine together two steps, the design step and an estimation step. For nonlinear models, as you all know, the variance covariance metric depends on unknown parameters, which we in principle do not know at all. And if our preliminary estimate is bad, we may find ourselves in a very poor shape. So what we're implementing for the adaptive approach, for the dose response examples, for each next cohort of patients, we will search for doses which are optimal given current parameter estimates. And this approach, which was not called adaptive but was called sequential earlier in the mainstream statistical literature, was introduced back in the 60s, probably in the paper by Box and Hunter. You can find the description of this sequential approach in Valeri's earlier book. The theoretical properties of this approach are not explored to the full extent. There are many, many open questions. Here there are only some references on this subject, and the theoretical problem of the convergence of adaptive procedure and some details are still open. So that's why we resort to simulations, what we are doing and what many other people who are involved in this are doing. As far as the estimation algorithm which we use in our tools, we implemented what we called combined iteratively reweighted least squares. So it's very similar to iteratively reweighted least squares, reweighted least squares algorithm, which is known for many years, but we add additional terms when we analyze the residuals. That's why we called it combined, and the details may be found in our paper with Valerian Communications of Statistics. And we proved that this algorithm is equivalent to maximum likelihood estimate. But it's an analog of weighted least squares. I already said that the key object is the information matrix. I won't go through details of derivation of the information matrix for our models. I'll just give you one single formula which is true for multivariate Gaussian variable. When you know the mean of your observation and the variance, there exists a closed form formula for the derivation of the information matrix of a single observation, and this observation may be k-dimensional. It may be a vector, and you can find the reference. Why I put it here is just to show you that very often the variance of observation depends on unknown parameters of the model, and this will be the case in all my examples. So it creates some additional technical steps which you have to overcome. My first example is about the study in patients with Alzheimer's disease. All details may be found in a paper which we published with Sam Miller in Journal of Biopharmaceutical Statistics. It was a phase 2A study. The endpoint was the percentage decrease of a particular biomarker compared to baseline. And the plot with which we started to play at the beginning was this simulated trajectory, what pharmacokineticists thought about the behavior of the endpoint. You immediately see that this curve resembles typical S-shaped curve or Emacs model, and the variability is high in the middle of the curve. That immediately suggested to use a Emacs model for our base background model. We used model with 3 parameters because for this example it was natural to assume that the baseline parameter, or E0, is equal to 0. It was the baseline was sort of subtracted. So this is a standard Emacs model. What is again quite standard that for the variance we use 2 different types of models. One with both are with additive and multiplicative component. In the first case, the variance increases with those. In the second case, it's largest in the middle at ED50. And we have not 3 response parameters, but actually we have 5 parameters, both response parameters and measurement error parameters. The goal of the study team was to find the dose ED90, which attains 90% of maximal response. So basically, you solve this equation for dose X, and you see that at the end you get this expression for the your target. And this is a nonlinear function of unknown parameters. If we want to minimize the variance of this function, we end up with a popular C optimality criterion. And for this example, we implemented both C and D optimality. There were many practical constraints for this problem because it was first time in human study, and we had to go very, very slow and start with very small cohort sizes. We always included placebo. There were always 2 patients per cohort. That was agreed with the clinical team. The starting doses were really very, very low, and they were expected to be non-pharmacologically active. We implemented the constrained dose escalation. So I just want to mention that very often when we analyze theoretical problem, we play with some hypothetical design regions. In this case, we have definitely the restriction on our design region. We cannot jump too far from the beginning because we have to start very, very slow, and our design region was cohort dependent. And we agreed on certain factors to increase the maximal dose for the next cohort. You could jump quite high when you didn't see too much effect at the beginning, and then we had to reduce this dose escalation factor for later cohorts when we observed some effect. Another practical constraint, we could select the same dose for all patients in a cohort, and this is again a practical restriction very often because you don't want to give different doses to different patients within the cohort, but in principle we had an option to select arbitrary doses for the same for patients in the same cohort. And we also introduced a standard design as an alternative because who knows what happens with the real study, and we also wanted to compare the adaptive design with very simple dose escalation design, more or less evenly spaced doses. So that, and then we compared operator characteristics of adaptive design and standard design and some other design. It's important to underline the following fact, and I think Valery Fedorov yesterday at his presentation mentioned that when you have multi-stage design and try to do some adaptation, the size of the first stage cannot be too small. If you have too few patients at the beginning and then try to run some sort of fancy estimation algorithms, you may find yourself in a very poor shape because your parameter estimates may be far from the true, and then it may take you much longer to get some sort of convergence to the true. On this screen, I give a screenshot of the MATLAB application which I created for this project. I'm not sure that you can see all details from the last rows, but basically it's a screen where the user can select different strategies for the simulation. The number of cohorts, the value of parameters, what kind of variance parameters the user wants, what kind of variance model, what kind of criterion DOC, whether doses are the same or not. And this is a screen which the user gets. There are options to create a single simulation or multiple simulation. I always recommend to start with single simulation because you can get the plots, you can get an idea how the algorithm works. But in real life, we are not interested in a single simulation. We have to run multiple simulations and create the empirical distribution either of parameter estimates or selected doses or some function of parameters like ED90 in this example. And that's what we observed. For this particular simulation, for the first 4 cohorts, we used forced escalation. If you remember, we start very slow. We cannot go to the top portion of the dose response curve from the beginning. So we have to really force doses at the beginning and not do any estimation. And we start adaptation from the 5th cohort, and this is the histograms for the selected doses for the 7th and 8th cohort. They were the last 2 cohorts. And if you look at the combined weight of these weights near the optimal doses are presented by these dotted lines. The combined weights are quite close to the optimal. For the locally optimal design, we have about 30%, 30%, and 40%. So we kind of see that if we collect more and more patients, that our selection of patients gets closer and closer to the optimal design. Operating characteristics. The important comparison is to compare efficiency of the design. For D efficiency, we take determinant and take root of order 1m. M is the number of unknown parameters. And usually the comparison is with the locally optimal design. For this example, the comparison with locally optimal design doesn't make much sense because remember, for the locally optimal design, we have the full design region from the very beginning, and we can select whichever doses we wish. For adaptive design, we always include placebo patients. We always include those patients which are forced on low doses. That's why the comparison adaptive versus locally optimal will be simply unfair, because adaptive design will have too many patients on, in a sense, uninformative doses. A much more reasonable comparison is with the composite design, or Valery called it a 2-stage design. What we start with? We start with this initial design, putting 16 patients on placebo, like here, 18 patients on low doses, exactly as adaptive. And then we allocate the remaining 24 patients in this example with respect to composite d-optimality. So this is the term from the initial design. This is the term from the design of the second stage, and you have to use this proper weight delta. And when we compare the efficiency, it turns out that the mean efficiency of adaptive design is close to 90%, which tells you that the adaptive design is able to recover about 90% of information only from 24 patients in this case, less than half. I think it's a pretty good performance in this case, and this criterion is exactly what's called Bayesian d-optimality in the classic mainstream design literature. You can check Valery's book or the survey paper by Catherine Challoner and Verdinelli in 1995. This is exactly the Bayesian optimality criterion which uses the prior information. We created, again, as kind of operator characteristic histograms of doses ED90, which were selected from 4 different methods: this fixed dose escalation, adaptive, composite, and locally optimal. You see that, again, Of course, locally optimal is the best, but when you compare composite and adaptive, the loss in mean squared error is actually not that big. And that's what it's about 10%. So again, adaptive design recovers the information rather quickly. My second example is about another study with a binary logistic model where the binary endpoint was of interest. One thing which we added to this model, the typical binary logistic is where the probability changes between 0 and 1. And in practice, you may think of situations that you cannot expect probability of success exactly 0 at low doses. It may be bounded from below, it may be bounded from above. So we introduced those lower and upper bound for the probability. The logistic model is really almost beaten to death. The results about optimal design are known for many, many years, and this is one of the papers going back to the 60s, which is not very often cited, where the first results about optimal design for binary logistic model were published. And we created a, again, similar standalone tool. For this example, we added the option of creating composite design from the beginning, those lower and upper limit. And again, in my first example, the drug was administered intravenously, so it was natural to assume that the dose range is continuous. Here, we introduce an option of using a discrete set of doses. When drug is administered orally, obviously you don't have a discrete continuous range, so the user can play whether the range is continuous or discrete. And another thing which we added for this example, we added not just the histograms, but empirical confidence interval, and 80 and 90% empirical confidence intervals. I show you for the parameters and for dose EDP, and what you see that the size of the empirical confidence interval goes smaller and smaller when you increase number of observations. That's what you expect from a reasonable estimation and design method, and that's the histograms are the results which you show to the clinicians as to what they may expect from the variability given the setting for the design. Third example is a very similar to the first, but we added the E0 parameter. It was a continuous logistic model. It was not unnatural to assume that it equals to 0. The peculiarity of this study was adding cutoff values at the top and at the bottom. We don't want to select doses which are too high, which give a response which may be close to the upper asymptote. And we don't want to give doses which may be too low and the response, the doses may be not efficacious. And another thing, we introduced what I called enrollment buffers. So when you don't collect information from all patients which are already enrolled, but you need to do some projections for the next cohort. And that's what those enrollment buffers are for. Composite design is a standard. We introduce those cutoff values, and the user obviously can play with different numbers and then explore the operating characteristics of a particular scenario. So these examples are along the same lines, and it was exactly the historical order how I worked on this project. These are examples of dose response. The second, how much time do I have? I started later.
Okay. 3 more minutes.
That's what I want, as I said, to bridge what we discussed last week with what we are discussing today. Optimal design for population PK/PD models. We have a workshop among those interested in nonlinear mixed effect models and developers of software for PK/PD called PODE, or PODE, Population Optimum Design of Experiments. The discussion started 4 years ago, continued for the past 4 years, including last week when we had a workshop here. We have 5 tools available, developed in France, and France Monterey is one of the main developers of that tool. Our tool, what is called PK Stamp, tool from Manchester, from Sweden in Uppsala, and Steve Duffel is developer of the tool from New Zealand. So 3 of the developers are still here. The typical model which we use for PK/PD is a mixed effect models. We have the response parameters. It's usually rate constants for PK/PD. We have individual parameters of individual patients which are sampled from the population, either normal or more realistically log normal. The data has both additive and proportional component of variability. We want actually to estimate the combined vector of parameters, the population mean, population covariance, and elements of measurements error vector. The simple example is a one-compartment model with linear elimination. You have seen this model many times in mainstream design literature. Our library is called PK-STEMP. This is where the name comes from. It was designed as a collection of independent modules created for various projects with GSK, PK, and PD modelers over the years. The last more than 2 years, I think it was a very successful and fruitful collaboration with my longtime friend and colleague from my Moscow institution, Alexander Aliyev. The screenshot of the typical application is here. You can select between different standard implemented modules, 1, 2 compartment with different types of administration. You can select parameters as random or fixed. You can select different type of population covariance. You can select different types of dosing regimens. There is a special screen how you select candidate sampling times to define the design region. And there is an option when you run and get the optimal design to validate, to run some sensitivity analysis to check how robust your optimal design is. We can address more complex models. The first one was the screen with a one compartment model which admits closed form solution. We can address Michaelis-Menten model, which is a system of nonlinear differential equation with this nonlinear term. We can address combined PK/PD models, where the input from PK here becomes output from PK, the concentration becomes an input into the PD. We can address cost-based design. It's the way we do it is actually simpler compared to what Valery discussed yesterday and Luc Pranzato discussed today. It's a simpler approach, but this is the critical paper, which is almost 60 years old, which introduced this approach. And finally, we can address really quite complex model. This is an example of a benchmark test. We have just run it and finished last week. So we have a PK component. It's a one compartment model, but with a constant infusion term. We have a PD model, a system of nonlinear equation. The PK component, the concentration, enters the PD component, and we were able to construct Fisher information metrics for this example, and at the end established that all 5 software tools give exactly the same results, which was really a nice validation of what we are doing. And you may take a look at this model. It's not that simple. So I think it was a very good example, particular for me. Again, doing this example, we introduced a user-defined option in our tool. So the user, without writing any code, can write down the equations, either in terms of right-hand size of differential equations or in terms of closed-form solutions. And finally, concluding remarks. I think that the most important things what optimal design do is in providing most informative solutions, either doses or sampling times. It's not at the end of the story, not the end of the story, of course, when we can estimate parameters as precisely as possible. There are much higher clinical goals, but this is a very important step. Another thing with optimal design, they give us a benchmark. We can compare alternative design, like in dose response, standard dose escalation compared to composite design or adaptive design. So we may find out that standard design perform almost as good as adaptive or locally optimal, and then we may use standard design because logistically it's much more simple. And another thing which is important, we may incorporate costs, but there were many talks about cost and utilities. So thank you very much.
Thank you very much. I think that the end was overwhelming. It's time to go for a tea break or coffee break. We will have an opportunity to come and discuss